What we do is we create the kind of diversity that we create this population of, say, organisms. Let's say for round numbers, it's 10 million organisms, right? We create that. We physically make those organisms. We do that using techniques like whole genome mutagenesis. We actually don't necessarily have to go in and engineer those changes one by one. We can actually just use random processes to create those mutations. And there are two advantages to working like that. Number one is it's fast. You can create millions of mutations by exposing a population to, say, just UV light. You'll get millions of mutations. So it's anywhere can be the solution.
Welcome to the Smart Biotech Scientist, the podcast that helps you master bioprocess CMC development and biologics manufacturing. I'm your host, David Brühlmann. Today, we're diving into the fascinating world of high-throughput screening and AI-driven bioprocess development with Jeremy Agresti, the founder and CTO of Triplebar Bio. Jeremy is a pioneer in microfluidics, so get ready to discover how Jeremy and his team are revolutionizing biologics development through their proprietary hyper-throughput screening platform. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist Podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Jeremy. Thank you for joining us today.
Great to be here, David.
Yeah, it's a pleasure. Jeremy, share something that you believe about bioprocess development that most people disagree with.
Yeah, I think I have a controversial take here. I think that most problems in scale up are not really problems with scale up. I think they're problems with the input. So I believe that if we had better, say, and if we're talking about microbial production or we're talking about pharmaceutical production, if we had better cell lines and strains, I don't think many of the problems of scale up would exist. Usually we think of scale up as being this difficult thing, and it's very expensive. And of course, there's a lot of science and knowledge that's behind it. But I think most of the problems we try to solve at scale could be solved at smaller scales before that we even get to that point. That's a big part of the impulse of starting my company was if we could engineer strains better, then we could design them for manufacturing. And then a lot of the problems in manufacturing just wouldn't be there.
Yeah, it's all about having the end in mind, right? And then knowing what you're doing to make better decisions along the way.
That's right. The way I think about it is like, let's try to solve problems when we have a lot of design freedom. And we have a lot more design freedom evolving or engineering traits in an organism than we do manipulating things like temperature and oxygen transfer and other kinds of processes in a large scale factory. So let's solve the problems early on when we can.
Absolutely. And I'm excited about today's discussion because we're going to look at how we can unlock biology's full potential. But before we talk about the science, I'd love to talk about yourself. Jeremy, can you paint us a picture of your professional journey that brought you to doing the work that you're doing today? And what were some interesting pit stops along the way?
I started as a biochemist, or geneticist really is what I studied as an undergraduate, biology. And I had a pit stop in a lab where I was studying evolutionary biology and population genetics. We were studying natural populations of fish and other aquatic species. I was very interested in that science, but maybe more motivated by the technology of how do you answer those kinds of questions. I think I realized that was one learning point at that part of my career was like, I like developing tools. That was an important seed that was planted. During my PhD, I joined a lab in Cambridge in the UK. The overall group was run by a scientist named Greg Winter, who had invented a technology called phage display, which is an incredibly important tool for antibody discovery and development. What was going on in the group that I joined, the kind of subgroup of Greg's team, was antibodies binding to a target are one function of biology. It's a very important and interesting one. But what are the other functions? Can we apply those same principles to other biological functions? So we worked a lot on enzymes and other kind of catalyst development. And that's where I was first exposed to this idea of miniaturization, because the way we solved that problem was to basically take the idea of a test tube, which is just a container of liquid that is isolated from other containers of liquid, or really the concept had existed in the lab before I got there. But they thought about this idea of what's the kind of quantum state of a test tube? What's the smallest test tube that's practical? That's probably the size of a cell is the smallest test tube that you could operate. You can't really put a biological thing in a smaller test tube than that. How do we make a test tube on the scale of picoliters? And the really cool insight was like, well, I can't move around drops of liquid mechanically very well in air, like a pipetter at picoliters. But what if I made a droplet of water surrounded by oil? That becomes this quantum size of biology. I can make a droplet of water the size of a cell. I can look for things like new enzymes. I can look at function. I can look at the properties of cells and manipulate those cellular scale. That was another kind of key learning point, was the concept of miniaturization. What's the container that you can go down to? We reached the end of what we could do as biologists. We were working with these, essentially a drop of water and oil. We commonly refer to that as an emulsion. It's a dispersion of two fluids that don't mix. And then my career took a change. I saw a talk by a scientist named Dave Waits at Harvard, who's a physicist studying emulsions. In his lab had developed tools how to manipulate emulsions in a really controlled way using microfluidics. So using patterned channels basically on a piece of glass, he had worked out how you can start to manipulate those droplets. So I joined his lab and really started to build within that physics team biotech applications for these nascent concepts of controlled biology and microfluidic emulsions. And so we worked a lot on how you would manipulate those emulsions to do all the manipulations you might do as a scientist in a test tube. Me being a practical person, and like I said, I'm interested in tools and their applications. I went to a company called Amyris. At the time, I think they would have called themselves a biofuels company, but today we might call them a synthetic biology company. And they were looking at how to engineer organisms to make diesel fuel replacements and other kinds of hydrocarbon replacements. And I think it was a great time for that field. I think what I learned by going to Amyris was when you have a big problem that you want to solve, you can attract this amazing high quality team to put on a problem. And I think the team at Amyris was super inspiring. When I left there, I went to a company called Biorad, which I think every biologist knows. It's a supplier of just about anything you can imagine for your biology research. And what I learned at Biorad was, I think I started to understand what products were really. And I had a lot of great interfaces with product managers who were bringing in customer needs to the R&D team and like what the world needed us to make and what the specs and requirements of those products were. And then really amazing like project management as well, and like understanding how teams get structured to meet goals. So I think all those things together, sort of the technology, the teams, the product focus came together when it was time to start Triplebar. All those pieces were sort of in place.
That's really exciting. I love your story, Jeremy. And now, yes, you founded your company. You've been in for several years already, and you are unlocking biology's full potential. Like, how do you do that? Can you tell us the 2-minute version, like really the high-level version of what you're doing?
I think it starts with the product in mind, right? The way I talk about it is we're a product-focused company that's enabled by our technology. So we start with where there's a product need. And so we've identified a few areas in both food and pharmaceuticals that we feel like there's a demand and a need in the world for product innovation. And then we kind of look at our technology stack, like what we have available to us, and we apply this technology to solve those problems. And the things that our technology are really well suited to are where, kind of like I described way back a few minutes ago in the phage display, it's places where we as a scientific community can't design the system. And so we need to be able to engineer a selection system to be able to let biology solve its own problems. We take a problem, say high yield production of a protein in yeast. That's the problem. The protein is something that is either difficult or expensive to make using, say, animal agriculture, for example. And then we try to engineer those yeasts to make high yields of those proteins. And we do that by basically testing a lot of variants of that organism using our miniaturized system. So we miniaturize these test tubes down to tiny droplets of water, and then we're able to screen millions, tens of millions. Sometimes if we look at the individual mutations in the genome, billions of mutations to try to find the solutions to those problems.
When you say screen, does this mean physically in the lab running an experiment, or is that using technology like AI or so?
It's a combination. What we do is we create the kind of diversity that we create this population of, say, organisms. Let's say for round numbers, it's 10 million organisms, right? We create that. We physically make those organisms. We do that using techniques like whole genome mutagenesis. We actually don't necessarily have to go in and engineer those changes one by one. We can actually just use random processes to create those mutations. And there are two advantages to working like that. Number one is it's fast. You can create millions of mutations by exposing a population to, say, just UV light. You'll get millions of mutations. It's not hindered by our knowledge of what we think our knowledge is of a mechanism of a solution. So it's anywhere can be the solution. And then we actually put them through our screening system, our wet lab screening system. We put those cells into, we call them microbioreactors, small containers of liquid that we're able to measure their properties. In this case, it might be measure how much protein they produce. And we're able to sort the things out that are high producers from the things that are low producers. And then this is where the kind of hybrid approach comes in. We can take the winners of that population and out of that population, and we can get information from them that then can be used for computational design to further optimize the system. So we want to know what's the performance of a particular genome. Like, the way you think about this genome, it's maybe a string of 10 million letters, right? And maybe 100 of them are different in each of those organisms. And we want to know which of those changes are associated with the higher performance. And so we can input millions of sequences into a database that we can use then to now use computational tools to make kind of design recommendations. So based on all of these outcomes, what are the changes that are important for a particular protein? And if you have enough data, then in the same way that you can have the kind of language-based AI tools that we're all starting to use now, you can have behaviors and recommendations out of a model that it wasn't specifically trained on, this transfer learning. That's also possible in the biology language as well. If you have enough data across enough proteins, you can start to predict, okay, now I have a new protein. How would I change that genome to make that protein? And so we use a combination of those tools to take in the data, create the training sets, and then design the next generation of improvement. And we've shown that in publication at the kind of protein level that we're able to use these kinds of tools trained on our data to make amazing progress in enzyme design, for example.
Now, as you're cultivating cells, the picoliter scale, and also you're using micro bioreactors, I imagine the conditions are very different than from the conditions you have at larger scale. So can you just tell us a bit like what are the differences and also how well your systems scale to a larger scale? Like once you have certain results, how predictive are these results for a larger scale?
It's a little unexpected for even me, like how this kind of has shaken out so far, this idea of scaling. I could have chosen this as my controversial opinion. It's a common just understanding that scaling up from different screening scale and the discovery scale to production scale, you get less and less predictive. And so the common wisdom would have said, gosh, if I go from a 1 mil screening scale and a 96 well plate to a 1 liter fermenter, I know that's not very predictive. So then you go down 6 orders of magnitude smaller for your discovery phase. It's going to be much worse, that connection, right? The correlation. We don't see that at all. I think we understand why. So first of all, like we've spent a lot of time, myself and others who have worked in this field of microfluidics, to kind of create a biocompatible interface for organisms at that scale. So as far as the organisms are concerned in this microscale, they're really they don't know that they're in a small volume. They just think they're in a tank. They don't feel the interface that they are growing as if they were in a tank. And so there's not really a difference there. And we see that in so many different ways. Like the assays that we can develop at a regular small scale, like a microliter scale or mil scale, just completely scale down. Once they work at that scale, we can get them to work at the microscale. It's that you have to know some things about how to develop that, but it's not fundamentally different. And then the other advantage that you have when you're working at very small scale is that the entire library that you have, the entire population can be in one tube. And you can also screen it in a day. So the cost of measuring everything in that population is relatively small each time. And so you can actually have situations where you understand, okay, here's my large scale process, here's my micro scale process. I'm going to test under more than one condition at the miniature scale, at the micro scale. So I can find things that say mutations that are beneficial at a high pH and a low pH, or a high pressure and a low pressure. And instead of picking your best estimate of the scale condition and testing under it, you can actually test under multiple conditions. And so you can find the robust solutions to the problem. And it would be really hard to do that at not miniaturized scale, because it costs so much money and time to get through just a few thousand variants that you never have a chance to test under multiple conditions. That's one way it scales better, is the ability to stimulate those conditions. We have something like 90% of the things that we pick out of our small scale. Ultimately, if we say they're better at that scale, they're also better at the kind of liter, 100 liter scale. And then once you get to a certain scale of production, then you can be pretty predictable. And so when we screen at the micro scale under conditions that are take into account the limitations of large scale, if you ultimately this is going to be in a 100 cubic meter vessel to be at production, we know there are certain oxygen limitations of those kinds of vessels. So we screen under those same limitations. So we're not finding things that can't be scaled up. We're screening for the scale process.
And how complex are your microbioreactor runs? Like when you're screening, is that a batch condition, or is that kind of a perfusion system at that scale, or how do you do that?
We often, for one campaign, will screen under different conditions, but we have ways of perfusion and feeding that we can apply to the microbioreactors. We can work in a batch condition. We can work in an equilibrium point where they're mid-feed, mid-growth, or we can work at like an endpoint. And the different traits that we acquire are specific to how we screen them. I think the quote is attributed to Francis Arnold, you get what you screen for. And that is what evolution is. You screen and then you get that. So we want to be able to manipulate the conditions to make sure we can get the thing we want. And sometimes it's cryptic. Sometimes I think biology, it will answer the question you ask. That's one reason we can work under multiple conditions, is to, just in case we were really asking a different question than we thought, we can still catch the solution.
And I imagine the system can be used for any kind of cell types. You mentioned yeast, but I imagine for mammalian cells, other cell types, or are there any limitations somewhere?
We use it across any number of cell types. So bacteria, yeast, mammalian cells. I think that a lot of really interesting biology is done with mammalian cells, which are larger and more delicate than microbes. And so we've worked with organisms like the kind of workhorse of biologics production and expressing biologics molecules in our system. We work with a number of other animal cells that are used in cultivated meat. So cells from pigs and cows and fish that work just fine in our system that you can grow and select for traits in the system. So it's pretty adaptable. I think the area that we've probably worked the least in that's relevant to bioprocess is filamentous fungi. We just found that working with single cell fungi like yeast, like Pichia, for example, like if you have evolution going for you, you can find single cell organisms that are desirable from a process optimization lens. You can find them that are as good as filamentous fungi for expressing heterologous protein.
That wraps up our conversation with Jeremy Agresti. We've explored how Triplebar Bio is transforming bioprocess development through miniaturization and advanced screening technologies. Join us in part 2, where we will dive deeper into the role of AI and data analytics in unlocking biology's full potential. And before we go, are you looking to accelerate your biologics development while keeping costs in check? Develop biologics better, faster, at a fraction of the cost with our fractional CTO services. If this is of interest, book a free call with me with the link in the show notes, and I'll see you on the call. All right, smart scientists. That's all for today on the Smart Biotech Scientist Podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, Visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.